Dear students, with this lecture, I continue describing the neural system. As you know, the goal of this lecture series is that you have a large number of sometimes apparently contradictory data in your head, and I like to select out, highlight out some important ones, neglecting some non-importance, and build a structure in your head, a system, what we can use well, you will not forget it and use well during your profession. Up to this point I described the autonomic nervous system and the somatomotoric system. Today's topic is the somatosensory system. As you know, the goal of the neural regeneration is that to collect information from the environment, analyzing it, deciding how can we react it in a way that it will be for a short and long run the best for us, and then behave accordingly. So they consisting of picking up information from the environment, these organs named the receptors, then sending into the central nervous system. This is named afferent or sensory pathway. This is synonyms in this extent. Then we process in the brain as a central processing, and whenever we decide, we send out to tissues which can respond to the brain. This is what we named efferent and motoric system. We name motoric even if it's not movement, but something like the gland secretion. And this can be somatic, which is we voluntarily do, and autonomic, which is we can do behind our wish, and the effectors, which can be three types of tissues only: skeletal muscle, glands, and smooth muscle. The motoric part was described previously. Now we see how the afferent sensory part works. We divide this one also in two parts: the somatosensory system, somatic system, the result of which we perceive, we feel, we know what's happening, and this can come from Sergoni organ with specific information, hearing, seeing, whatever, and diffuse from receptors spread all over the spleen or located in the muscles and tendons, which gives another information. In contrast, beside the somatic, we have autonomic, visceral sensory part, which is not perceived, and this is a part of the autonomic system. Today, I will talk about this area, the somatosensory system. The somatosensory system, the first step is the receptors. Receptors are not part of the neurons, but these are a kind of service organs, these sensory epithelials, most of them, and this will process the information, translate the information to the language of the neurons, nerve cells, and then the nerve cells picks it up and processes it. The receptors themselves can be divided into different subgroups. Exteroceptors. Exteroreceptors are those receptors which picking up signals from the external environment, what's happening around our body. These can be teloreceptors. These working with information which has no contact with us, provided by Sergoni organs like seeing, hearing, balancing, and so on. We don't have to be in contact with a person to see or to hear him or her. We have contact receptors in contrast. If we can analyze signals only if we are in physical contact with that, like touching, temperature, smoothness, and so on. They are mostly located in our skin. Beside the exteroceptors, we have the interoceptors. These dealing with internal signals, such as signals is coming from inside the body. We have a very large variety of these signals, physical, like temperature, pressure, tension, and so on, or chemical, concentration of many different chemicals like glucose, oxygen, carbon monoxide, and dioxide, and so on. And finally, we have a third group of receptors named proprioceptors. Be careful, this expression used for many different purposes, even in the lectures of the same department. in our meaning, in my meaning, it means a group of receptors which actually processing internal information, but we can also conclude external conclusion. We can draw external conclusion. For instance, the muscle spindle is among them. The muscle spindle actually feels stretching of a muscle, which is very well used for an internal processing of the motoric center, as I described before. But at the same time, we are aware of this tension, and we can judge weight, how heavy this stuff is. Similarly, stretch receptors, in the Ruffini receptors in the capsule of the joint will exactly tell you which way the capsule is stretched and exactly which position of the limb is. If you feel about 2 degrees of the position, even without seeing anything. So internal information can have an external conclusion. This is what, in this meaning, we have proprioceptors. The sensation, the quality of the sensation also differs. We have both phylogenetically and anatomically and functionally 2 different groups of sensations. The one is the proteopathic. The name describes proto means elementary, pathos is Greek for feeling. So these are the fundamental sensation. It means it's very important for you. If you don't have that, we have an immediate danger or frequent danger of being injured or dead. The main feeling in such a group is the pain. Those creatures, animal type of creatures who do not feel pain very soon become extinct because they simply don't feel that one part of our body they damaged, and they not only can lose that part, but for instance the infection can propagate and the entire body is dies, disappears. Consequently, the pain is one of the most elementary, most fundamental feeling. Also, we have 2 other group belonging to this group. The extreme temperature, freezing and burning, is also damage, completely kills the various body parts if it goes on. And the crude touch. Crude touch or superficial touch means that I have a feeling that something is in contact with our body. This is not necessarily dangerous. So we have to decide with other means what is the significance of this feeling. It can be dangerous, like a spider or a snake is climbing out on our back. It can be very pleasant if a girlfriend touching us, or quite neutral, just a pillow is in contact with our body. So by itself is not an alarm signal, but you better watch out, calls out attention that it can be also potentially dangerous. These signals are processed by a long developed primitive nervous system, and this is also present in our body. Unlike the politicians, the biology works in a principle that if everything proves to be good, we do not discard it, but use it as long as we can. And similarly, this pain processing was present in the same way milliards years ago, and practically in the very same way we use it, and very successfully. The epicritic sensation quality is for the fine discrimination. It employs large nerve cells, thick fibers, which has very high energy consumption and needs space. So only creature who can afford that utilize this. So this is a latest development of the phylogeny. Among them belongs the fine touch. We see the surface, whether it's smooth or rough, the softness of the surface. We feel slight temperature differences. For instance, if you take as object in my hand, depending on how much temperature is drawn from my hand, I can differentiate metal, wood, plastic, and so on. Just a very slight differences, the apparent feeling of temperature. The vibration is a very quick variations of pressure. It also requires an epicritic type of information, and a very fine detection of stretching, very exact measuring how much is stretched. This also belongs to that. Now, this is after this general introduction, I try to describe in details, anatomical details of these two sensory systems. The proteopathic systems start with proteopathic receptors. As I mentioned, most of the receptors are epitel, sensory epitel. This is one of the exceptions because the pain is felt by no external component, by specialized membrane part of nerve cells. So do not require an external cells for that. We have a TRP ion channels which are sensitive for certain mechanical and tough chemical information. Further details you study in physiology and biochemistry. The other receptors, the temperature and the touch, are simple skin receptors, rather the Krause ending or the Merkel touch receptors can do that. The peripheral part of the proteopathic system is located in the same way as the other, the epicritic, but morphologically look different. In the sensory ganglion, you can see large nerve cells and small nerve cells. And those nerve cells which are working with the proteopathic system are very small. You know the general rules. The pericardial nerve cells is nothing else, just a kitchen. Whatever it requires the maintaining and feeding the most important part of the neuron, which is practically the membrane. All the functions which is necessary for a neuron is related to membrane. In the membrane, the receptor, then the axion potential generated, axion potential propagates to the membrane, and through membrane in the synapses, through the membrane, the synaptic vesicles, the neurotransmitters are given off. The size of the body is proportional to the size of the axon. If we have thick axon, which is necessary for the fast, reliable conduction, this can be fed by a big kitchen, a big pericardion. If the axon is thin, by the way, slow conducting and relatively primitive, small pericardion can feed that. So the smallest pericardia in the sensory ganglion working in this proteopathic system. These are located as usually both the spinal and the cranial, the ganglion of spinal and cranial nerves. In the peripheral nerves, the axons are the thinnest, as I mentioned. Some of these, especially the pain, can be unmyelinated. This also has a kind of theoretical importance because they're conducting very slow. We really feel it. For instance, if we drop something on our foot and we feel pain, it takes a very sensible, about 1 second until the damage happens, we see it, and we start feeling the pain because it takes practically almost a second until this information reaches our brain. A similar problem, some people think that one of the reasons why the dinosaurs died out is because they have only proteopathic system. And let me say, if the tail of a 30-meter long dinosaurs is damaged, it took, as they calculated, 3 minutes until the information reaches the head. During that time, either the tail was chewed off by somebody or burned off or so. So this type of nervous system is not really practical. This is one of the reasons why they died out. Central nervous system, whenever the fibers, the stem fibers, arrive at the spinal cord as the spinal region, immediately terminates as rexed lamina number 2 and 3 in the posterior horn. In the brainstem, we have a single nuclei which any kind of cranial nerve which has pain sensory fibers arrive at that. This is named the nucleus tractus spinalis nervi trigemini. This is what I will talk about in details later on. Now let's see the spinal part first. First, this is what we name the Spinothalamic System. This is a system which forwards the pain signals from the spinal cord. The first step is a very large number of nerve cells. This is the largest number of nerve cells. This is the far the biggest computing potential in the spinal cord. This is what we name substantia gelatinosa. It's lamina number 2 and 3 by Rexed. If you see in Birchhoff-Stein slide, when you see the cell bodies are yellow and the fibers are dark brown, this area is yellow because it's mostly cell bodies. The axons are short, even although they're rich, they are short, so it's mostly that. The similar way is named substantia gelatinosa. Whenever we illuminate this area, unlike the white matter and all the other area where we have a lot of fibers, myelinated fibers, we have a total reflection of the light, and we see this area white because the light behind us reflecting to our eye. Here we have cell bodies. The light can go deep into the cells because it's transparent and they're losing a lot of lights. Then when it reflects, it's become much darker, a little bit translucent, a little bit jelly-like. This is why it's named substantia gelatinosa, because microscopically close to the jelly. In the rectal lamina number 1, we have also nerve cells, which is working in this topic, and this is relatively bigger cells, and the axons go up and down in the area wherever we have the posterior rootlets, and this zone where these fibers are, the Lissoyer zone. This makes a communication between the neighborhood segments. The reason for that will be explained soon. This system is one of the most responsible systems in our body because it should be very carefully worked. Probably you have everyday experience that the pain is a very aggressive feeling. Whenever you feel real pain, you are forced to stop any activity, doesn't matter how you like it, and you will concentrate on your injured body part. Any kind of false alarm would make our life much more difficult. It practically in every second several times happens that some of the pain receptors just don't feel good. It's a kind of renegade fibers and sending signals up to the spinal cord. These guys, these little cells, must evaluate whether it has a real damage or something non-significant things, very little problem, but can be solved locally. We don't disturb the brain, and it will not forward. This is a kind of graph which shows that if this is an incoming signal coming from the receptors, and this is outgoing signal from the system goes up to the brain, first, whenever the incoming signal starts increasing, it doesn't grow parallel, but it's suppress the outgoing signal. Do not feel everywhere pain, just let me work. The problem will last locally. Whenever the incoming signal reaches a certain threshold, it not only passes through the signal, but also amplifies that. And this is also relatively sudden. And this is also has a consequence. Not only do not disturb us, the small signal, but the pain usually appears quick. For instance, if we have a toothache, most of the time the pain comes suddenly. The process behind the scene is going on for days, but whenever they reach a certain level, these guys think, oh, that's serious, and forwarding suddenly the whole signal. And this sudden onset of the signal is also alarming, and this is also serves the final purpose. Whenever these little guys in the substantia gelatinosa decided that it's really serious, this information is taken up by cells, usually oval type of cells, in rexed lamina number 4. and the axons of them crossing and going up on the opposite side in the borderline between the anterior and the lateral funiculus all the way till the thalamus. This is what we name the Spinothalamic Tract, named after Edinger. The Spinothermic Tract is very interesting things. Many general rules of the nervous control can be described. For instance, do you know the Calais rule? Whenever fibers is longer, going up or coming out from the lower limb is located further away from the gray matter. Whatever is shorter, closer. Also, you can see this shed of the color showing this comes from the neck, body, or the lower limb inside the spinothalamic tract. Also, we have this is what we name the somatotopic localization. Also, we have a functiotopic localization, different area of the spinothalamic tract dealing with different function. These guys forwarding the extreme heat, these guys the pain, and these guys the crude touch. Some books describe the spinothalamic tract as the lateral and the anterior. Well, this is a very consequence of very superficial viewing things. If you can see a real slide, here, even although it's coming out fibers of the anterior rootlets, but between the fibers we still have vertical running, cross sections of vertical running fibers. So practically the spinothalamic tract is continuous, just crossing fibers there. So some people who describe this book practically neglecting this, the most valuable part of the spinothalamic tract, whatever conducting tail. Even although some books refers anterior and lateral, so on the exam you better refer that, but you know that this is really a continuous pathway, which in the middle crossed by the outgoing fibers of the anterior rootlets. This, whatever described, was only valid for the pain. The other two, protopatic moiety, the temperature and the superficial touch, is far not as influencing our life, so it's not necessary to have to be that carefully processed. This is why the processing is very simple. With 1-2 cross sections, in reality we have 1 or 2 switching neurons, it's a little bit located dorsally. This is where it does. So these blue guys are also protopatic sensory fibers, but with a very low processing, the cells, output cells, also sending the fibers up to the spinothalamic tract. So whatever is important is the very careful processing of the pain, but the other two protopatic signal is actually a little bit more superficial. What happens with the spinothalamic tract? At the spinal cord level, this is elongated arch-like tract from the anterior to the lateral funiculus. This become a nice round unified tract, and this way it's passing through the brainstem. Both the medulla oblongata and the pons and the mesencephalon, it runs in the middle of one side of that as a single unit of fibers. Finally, the fibers arrived at the thalamus, so they do nothing in the brainstem, just they going to the thalamus. The thalamus is an interesting structure. A lot of students are afraid of them because it has many nuclei. If you consider this way, it's much more simple. Practically all of the nuclei of the thalamus, except for 3, working with processing sensory information. So it's a big sensory center. The 3 nuclei, 2 of them is the ventral anterior and ventrolateral. These are motoric, parts of the extrapyramidal system, as I described in the previous lecture. And the pulvinar is an interesting nucleus. It has only cortico-cortical connection, no subcortical, and this is different. The rest of the nuclei, all of them working with specific sensory, and most of them will be described repeatedly today. The information from the spinal area or also from the cranial nerves arrives at these 2 nuclei, the ventral posterolateral and posteromedial. Actually, the information has a very strong somatotopy, like an onion ring, segment by segment. The highest segments is in the medial one, in the tail segments, the lateral one. Practically it's a continuous nucleus. We named medial and lateral, just describing that whatever information comes from the cranial nerves, this medial part is named the medial one. Whatever arises from the spinal cord area named lateral. Otherwise, the structure and the function identical. The cranial nerves has also a very important detail. You have a question card on the final exam which titled something like that, the sensory nuclei of the brainstem. The first sentence you have to say if you really want to pass, and unfortunately most of the students will not pass on that, but generally, theoretically it's very simple, that unlike the motoric cranial nerves, those nerves which motoric function as well, the motoric nuclei, every single cranial nerve has its own motoric nuclei, named after the nerve usually. The sensory nuclei do not belong to cranial nerve, but belong to function. All the proteopathic information arrived at this violet nucleus. This is the nucleus tractus spinalis nervi trigemini, and the epicritic to these two nuclei, which I will describe later on. So the nucleus tractus spinalis nervi trigemini is the nucleus for all of the proteopathic information. One important thing to know is this is a direct continuation of the substantia gelatinosa. It's not visible borderline. The cells look exactly like each other, and also the function is identical. Moreover, even although this nucleus, long nucleus named after the trigeminal nerve, any cranial nerve which has pain, proteopathic sensory fibers, will send the information to this nucleus. For instance, if we have a fish bone in our throat, through the glossopharyngeal nerve, it goes to this nucleus. If you feel pain in our larynx, through the vagus nerve goes here, and so on. So this nucleus, even although it's named after trigeminus, is a common proteopathic nuclei. This is an extension of the substantia gelatinosa and has the same function. Because most of the fibers come through the trigeminal nerve, it is discovered with this connection first, and this is why it was named after that. After the information is processed in the nucleus tractus spinalis nervi trigemini, it behaves very similarly as the Spinothermic Tract. It goes to the other side and goes up all the way till the specific sensory nuclei of the thalamus, the VPL, in this case the VPM. This pathway is not named spinothalamic but trigeminal lemniscus, lemniscus trigeminalis, but this is just a name. As I mentioned, they arrive at the posteromedial nuclei, and the principle was described just before. Post-thalamic pathway. So whenever the information arrives at the thalamus, and that's very important to know that both the proteopathic and the epicritic meet each other, arise at these two nuclei, VPL and VPM. The difference is not the sensory quality from that point on, but the localization, body localization. From that, the information goes in three different ways. This is also a little mess up in the books, so I try to simplify this one. The one of the outlet is the superior thalamic radiation or middle thalamic radiation. This can be used synonym, refers to the same thing. And passing in the internal capsule behind the knee, behind the pyramidal pathway. The source cells are in these two nuclei of the thalamus, the dorsolateral and lateral posterior nuclei. So the information from the specific sensory nuclei, VPL and VPM, go here. And these nuclei just selecting out the information, what do they need. And this is the topology, where, at which point in a body something happened. So whatever pathway is in the middle thalamic radiation, the cell bodies, the pellicardians, is sitting in these two nuclei. The end of the axon is the postcentral gyrus. This is you get several details on the somatotopy of the postcentral gyrus, but very important to know that only thing they do is the localizing the source of the information. They do not contain any information on what type of feeling is that. What kind of attitude we can go with this information, just where, at which point something happened. The other output of the thalamus is the anterior thalamic radiation. Anterior thalamic radiation goes almost horizontally, so it's below the level of the flexic cut. It's practically not visible in the flexic cuts. And these coming from the medial in the anterior nuclei, also for the reticular nuclei of the thalamus. These nuclei have a very rich connection with the limbic system, and this is why some of the books pretty much mess up the function of these nuclei. It's really communicating with the limbic system, but in our view, in our extent, the important function of them is forwarding the quality of the sensation to the frontal lobe. The fibers of these pathways arrived at the tip of the frontal lobe, and this gives us information whether this feeling, which is localized in the postcentral gyrus, how can we, what does it mean for us? It is pleasant, so we can continue as long as we can. It's unpleasant, so we get rid of it as quick as we can. Or it's neutral, just a piece of information you don't have to mind, just know it. Some kind of medical experiences, medical practice has a very nice proof for that. There was a neurosurgery operation named Frontal Eukartomy, in which these pathways were cut, the anterior thalamic radiation. Nowadays it's very rarely done, but still done, but earlier it was done very frequently. It was done in two cases. First of all, terminal cancer patients in which they have terrible, intolerable pain, and all of the painkillers was not really effective. The morphine should be administered in so high concentration they would stop breathing. In this case, in this patient, whenever these pathways were cut, has a very interesting characteristics. So they tell, oh, it hurts here, I feel a pain, but who minds it? It is really not bad for me. I just know that something here happens, but absolutely doesn't disturb me. The bad news is that also don't have any kind of good feeling. So all the feelings suddenly become neutral. So the attitude background, what does it mean, this feeling, was completely separated from that, but still the localization was very good. The other group of patients who underwent this type of operation is the mental ill people who were very aggressive. With a very little disturbing, they break everything, wanted to kill around, and couldn't be treated in any other way. After this pathway was cut, they become calm, relaxed, nothing disturbed. They did not feel anything bad. Well, did not feel anything good as well. But most important was that. Nowadays, a couple of psychic drug can do these things, chemically paralyze, cut, almost specifically this pathway. So this is why the operation is lower. Probably some of you saw the film. It was done quite a good time ago. The One Flows Over the Cuckoo's Net with Jack Nicholson. If you didn't see, it's worth seeing that. Jack Nicholson played a very cheerful and very active people, and the environment couldn't tolerate that. They were sent to a hospital, and finally this type of operation was done that. And Jack Nicholson played very well these things. It was absolutely dull, nothing disturbed, no sadness, no cheers, nothing. It was absolutely neutral for the environment. It's became a very dull people. The third output beside the middle thalamic radiation and the anterior thalamic radiation is the direction of the hypothalamus. These medial nuclei and some of the intralaminar nuclei send information down to the hypothalamus. The hypothalamus, the main pathway is the mammillothalamic fasciculus. As you know, the hypothalamus is a center of all the autonomic activity. So this is a kind of information how the feelings will affect the autonomic functions. For instance, if in a certain area of the skin we feel high temperature, with sweating to cool it, also the vessels dilated to lose more heat. If it's cold, we have the hairs erected because the arrector pili is activated on that. Also, if we have terrible pain, sometimes it happens that sphincter getting lost and we are in a big trouble. And also the heart rate changes with, in the consequence of the feelings. This was the general idea on the proteopathic system and little introduction of the post-thalamic sensory pathways. In the next chapter of this lecture, I will talk about a kind of connections, site services of the proteopathic system. Whatever is already briefly analyzed before, there is a noise filter in the substantia gelatinosa, which aiming at not disturbing us with unnecessary things. Substantia gelatinosa is a very rich connection among this, a lot of cells, a lot of connection between them, as incoming fibers end up in many cells, and each cells get fibers from many receptors. This is why I can very carefully analyze whatever I described before. In lamina number 1, these big cells sending up and down the fibers in the least layer zone make an intersegmental communication. So if these cells feel that only I got some information, this is negligible. But if in the neighborhood segments also the same time we get bad signals, something happens and we can forward this signal. The other thing is that from the epicritic fibers, we also have collaterals to that, just helping these signals. So whenever we have signals from the pain receptors, which requires a very tough signal because it's very less sensitive, and do not get enough information from the epicritic, which is very sensitive, it means that we have a false alarm, so better just not to forward this information. The nucleus tractus spinalis nervi, the trigemini, works the very same way. One of the other services is the spinal reflexes. We have the Dosiceptive Reflex, also named flexor crossed extensor reflex. Usually it's given in a different lecture, and because it's not connected to the Spinothermic system, it's usually not easy to understand. But the whole reflex is very simple if you know how the Spinothermic system works. Let's see the blue fibers only. We step into a nail. The pain signal goes here, processed in substantia gelatinosa, and forwarded in the usual way to the thalamus in the Spinothalamic tract. This is the backbone of the system. However, with two collaterals, we have a very nice service for that. First of all, this is the same axon coming from the fourth rexed lamina, has a collaterals locally on the same area, and with the switching neurons, it will activate motoneurons. Because when we are stepping into a nail, this is processed by the sacral segment. We are already in the sacral segment, and the typical muscles of the sacral segments are the hamstring muscles, the flexors of the thigh. Whenever the pain arises here, we automatically contract this one. The consequence, lift up the leg from that. This is very useful for this shortcut, but as I mentioned, at least 1 second is needed until the information goes up to the brain. Then we start thinking, oh, what's happening? How can we protect? And when we move out the leg, it's already has a very big burning of cigarette whenever we stepped in. This makes a shortcut and remove even before they feel a pain. Probably you already recognize that when you make a bad step, for instance, your leg slipped down of the curb, you jumped a little, and then in about a second you start feeling pain. This is the shortcut. Additionally, the same axon has another collateral a few segments above. This is already a lumbar segment, and through the switching neuron, the same side will activate motor neurons. Because we are the lumbar segment, this is the muscle innervated by the lumbar plexus, the extensors of the leg, and this becomes stretched. Why we needed that? Whenever we lift up this injured leg, whatever carried half of the body weight, all the body weight gets to there. And if suddenly there is double burden, it will collapse. Preventing this collapsion is automatically contract this muscle, preventing the collapsion. Once again, this is a very quick reflex, and by the time the information goes up to the brain, everything is already settled. So it's very useful. And it doesn't need anything else, just two little collaterals from the Spinothalamic Tract. And if you really understand that, you will understand how this reflex works. So this is the accessory service of the Spinothermic Tract. We also have diffuse connection to non-specific areas. As you know, the pain signals, the signals for the damage of the internal organs is not feelable. We don't feel, it's not apperceptive. However, the pain signals from the internal organs is processed in the same area as the peripheral, the somatic pain in the substantia gelatinosa. And as you know, there is a very rich connection between these two cells and can be a crosstalk between these cells. If there is a pain from a certain organ, it's irradiating to the pain of the cells of the same segment, and we can have a kind of phantom feeling on the same area. This can be close to the position of the organ, but can be very far away. A typical example is the gallbladder. If you have a gallbladder cramp, you start pain first in the right shoulder. Why? Because during embryology, as you know from that, the diaphragm started developing somewhere here, the cervical region, because we did not have lung and heart at that time. Whenever the lung and heart developed, it pushed down the diaphragm there, together with the gallbladder, but the innervation remained at the cervical segments. And whatever the lower segment or middle cervical spinal nerve innervate, they feel the pain in this area. Much more interesting, the heart. The sensory innervation in the heart goes up to the cervical segment, the same area where the ulnar nerve have. If we have a damage of the heart, cardiac infection and so on, this pain goes to the eighth cervical segment, and we have a crosstalk, we have a phantom feeling, as if we had pain wherever the ulnar nerve innervates. And now the typical path of the ulnar nerve is in the left side is the fourth finger, left forefinger. And for several thousand years, the human observation already realized there is some connection between the heart and the left forefinger. This is why we're wearing the wedding ring in that finger. The rest of them you can figure out. These areas where we have the apparent phantom feeling, the damage of the organs, they named head zones. On the reticular formation is also connected to the proteopathic system because a very important signal from the air myotomy signal is the pain. How they process, what do we do that, I described in details in the previous lectures. So the proteopathic signals is one of the sources for the general activation done by the reticular formation. No, that was about the proteopathic system. Let's go to the epicritic system. Once again, the epicritic system is a later development of the phylogeny, especially mammals and birds have that. And these are much quicker reacting, much more delicate, much more little differences we can feel on that. The receptors are specialized skin receptors. The most typical is the Pacinian corpuscle. This is the most sensitive receptor, but if we have now from the previous lectures, these we have flat cells, large number of them. the membranes of which we have stretch-sensitive ion channels, and whenever we have slight modification of the shape of the cell, there will be a voltage difference between the two parts, and we have many cells. They are added together. It's a very strong signal amplifier, so the very little pressure can be felt by that. It's also recovering fast, so the vibration is also very well felt that. Also we have these Meissner receptors, and also this is the end of the Ruffini receptors, which also serves this information. Very typical is the muscle and the tendon spindle. The details was described before in the other lectures or in the practices. So the receptors are very sensitive, and they can give a very delicate grade of the signal. Periphery, they're conducting in thick axons. The biggest axon, the thickest axon in the peripheral nerve belong to the epicritic system. These are also conducting fast. Based on the information I gave before, the largest nerve cells, the largest nerve cell bodies in the sensory ganglion belong to the epicritic system, since this thick axon with a big volume requires the big kitchen to feed up, and as you know, the pericardium is practically a kitchen of a neuron. This is located in the spinal and the trigeminal ganglion. What's in the central nervous system? Whenever the epicritic system started developing, the body figured out, I need more sensitive things. Already the spinal cord was full taken, as there's no place to receive this kind of epicritic information. This is why the main body of the fibers try to find receptive cells and couldn't find anything until it arrived at the brainstem. So the primary nuclei of the main axon is on the brainstem. It doesn't not contradictory. It has several collaterals. We'll describe some later on, which may process signals and add additional signals. But the main pathway of the axon will not terminate in the spinal cord, but goes all the way up to the medulla oblongata. In the medulla oblongata, they arrived at the dorsal side in the closed part of the medulla oblongata, and the two pathway, which named Gold and Burdak pathway, arrived at here. Burdak pathway, fasciculi cuneati, is carrying signals from the upper limb, and the Gold from the lower limb. Obeying the Carle's rule, whatever is shorter, Burdak is shorter, is closer to the gray matter, in this case laterally. Whatever coming from the lower limb is pushed further away from the gray matter. This is why it's good medially. They arrived at the pair of the nuclei with the same name, nucleus fasciculi cuneati or gracilis, also named Gold and Burdak nuclei. From that, the information goes in the opposite side and going up to the thalamus, and this pathway is named the lemniscus medialis. As I mentioned before, they arrived at the very same nuclei of the thalamus, where the protopatic information arrived. This is the VPM, because this area from the spinal cord is on the lateral side. This arrived in the VPL. Whatever happening further on is identical whatever I described before, and this is going to the cortex in three different ways, and also to the hypothalamus. In the cranial nerve, as I mentioned, the continuation of the substantia gelatinosa dealing, the nucleus tractus spinalis nervitigemini, dealing with the protopathic information, and these two other nuclei dealing with the epicritic information. The primary is the nucleosensory princeps nervitigemini. This is why all the epicritic information arrives. Doesn't matter to which cranial nerve arrives such an information, it's arrived at this nucleus. For instance, if we have muscle spindles in the throat, through the glossopharyngeal nerve, this epicritic information arrives at this nucleus. Also, even although they named after trigeminus, it serves all the cranial nerves which has epicritic fibers. The nucleus tractus mesencephalicus nervi trigemini is a very strange thing. This is practically can be considered as a misplaced ganglion. Very uniquely, it has pseudoinopellar cells inside, so it has the same function as the trigeminal ganglion. The peripheral axon of these pseudoinopellar cells come from the muscle spindles of the chewing muscles, and the central ones terminate at the nucleus tractus, nucleus centris princeps nervi trigemini. The rest is the trigeminal lemniscus, a very similar pathway as came from the nucleus tractus trigemini, the tractus spinalis nervi trigemini, and as I mentioned, arrived at the VPM ventral posterior medial nucleus of the thalamus. Finally, we have a kind of cross services also, any kind of connections of the epicritic system. So this is the main body, muscle stretch receptors, and the dorsal side go all the way up till the Kohlen-Burdak nuclei. It has many collaterals at the spinal cord level. One of the collaterals ending at the motoneurons in the exatizine fibers where the muscle spindle is. Was a consequence of them. It's done frequently by the doctors. If you hit the patellary ligament, we stretch the muscle, and this information, stretching information, beside going up to the brain, this same information goes to the neuron, the motoneuron, and makes the muscle contract. This is not done just to have the doctors to play with, but it's very useful for us. Let's imagine we jumping down from somewhere. During the time we are in the air, the lower limbs has nothing to do. The muscles is completely relaxed. At the moment when we reach the ground, it's suddenly they have to resist the ground, so muscle has to contract. If we had no collateral here, the information went up to the brain. It takes time, even if it's quick. We have to think about, we reached the ground, what should we do? We have to contract the muscle. And by the time the information goes back to the muscle, already our nose is bleeding because we collapsed. This reflex does the same thing until the muscle is not stretched, until it's in the air, the muscle spindle is relaxed. At the very moment when we reach the ground, it becomes stretched. This information goes very quickly to the motoneurons. In the quarter of a millisecond, the synapses time and going back, and very quickly we contract the muscles even before we think about that. So this is also a very useful service, and this is the proprioceptive or stretch reflex. And functionally, we can consider it's an accessory service of the dorsal fasciculus, which is a part of the epicritic system. Also, as I mentioned, it can sense collaterals through the substantia gallitinosa, and this is kind of side information from the pain analysis. Also very important, maybe even more important, the cerebellar input. This system is pretty much confused in both the lectures in the books, and sometimes they proprioceptive things and so on. Proprioceptive is a reflex I've described before, and not that. What really is the following. Also a side service of the epicritic system. From the muscle spindle, the information goes up to the main backbone of the system, but collaterals going to nerve cells located in the stem of the dorsal horn. And from these cells, information go up to the cerebellum. Why? Because the extrapyramidal system, as I described in the previous lecture, need specific information, how the things going on. As the control of the movement, the extrapyramidal system sends out neural stimuli, and the muscle responds it in a very different way, depending on many factors. And this feedback must give information on how the muscles responded to signals. So this information go here. These nerve cells just selecting out the information, whatever is necessary for the cerebellum, and sending up the cerebellum. There is two ways they're doing that. In the upper part of the body, C4 to L3, we have a group of nerve cells very close to each other. We can see a nucleus structure. This is what we named Clark's column. From this, the axons going to the posterior lateral part of the lateral funiculus. This is what we name the posterior spinocerebellar tract or flexic tract, and go up to the cerebellum. In the lower part of the body, doing similar thing, this is the cells are diffuse, do not form any nucleus type things. This is why they do not have any specific name of that, but they, interestingly enough, crossing to the opposite side and going up to the anterior part of the lateral part of the anterior folliculus. It's very interesting to note, probably you remember from the previous lecture, that the cerebellum works on the identical side of muscle. The crossing is in the pons, so you know the one side of the brain controls the opposite muscle, but this crossing in the extrapyramidal system happens in the pons. Already the hemispheres of the cerebellum is ipsilateral, so it works on the muscles of the same size. This fiber behave very nicely because they give information up on the same side. So this is the kind of principle of the entire pathway. The upper part, the Clark's column, is going up, and through the inferior cerebellar pedunculus goes to the identical hemisphere of the cerebellum. What happens with the other way? The other things are a little bit complicated, tricky way. Very same way, cells processing the signals around the stem of the posterior horn, but they crossing, and in the opposite side they going up, passing through the medulla oblongata, passing through the pons, and arriving at the mesencephalon. And interestingly enough, they are crossing back. In this way, they also arrived at the same size as the signal source is. This is what it makes the information understandable. Probably the reason for that, for some reason, is that at the same size, some of the fibers terminate at the olivary nucleus, and this is what we name the spino-olivary tract, and this is also belong to the system. Maybe at opposite side information is collected to the inferior olive. This is also a little bit overcomplicating this tract in some of the books. This spino-olivary tract is actually a part of the anterior spinocerebellar tract. And here you can see, as in the previous lecture, the complex extramedullary system, and this system, whatever described right now, is this part of that. And this is what I wanted to talk about today. Thank you very much for your attention.